So, so all things are made up of cells. Some organisms are just one cell big. Like for instance, there are cells that are organisms that live in lakes. Amoeba and paramecium are examples of that are free living and they're only one cell big. Whereas we are obviously multicellular, many cells big. And so the cell is the smallest unit of matter that is considered alive. And all cells are related by their descent from earlier cells. So every cell comes, what we say, comes from what we call a pre-existing cell. So how does that happen? Those cells come from a pre-existing cell. Grayson?
They split. It like to get big and then they get so big at one point they have to like split up.
That's exactly right. All right, and so so they divide into two, they split into two, and then instead of one cell, they become two. And so a unicellular organism, that's how they replicate. That's how they get more of them. So a unicellular organism will grow and then divide, and now you've got two of them. When our cells replicate, we are not just one cell big, we're many cells big. When our cells divide, that's how we grow. We grow, we become more cells big. All right, and so therefore, as you get older, cell division and you stop growing, your cell division slows down. Okay, or else we'd be giants. All right, so that's not the case. And so also cells can differ from one another, but share also common features. And so this picture here illustrates that. You good? All right, this picture here is two different types of cells. This is the tail end of a neuron. A neuron kind of looks like this. It has a cell body with little features that come out here, and then they have a long, what's kind of like a tail, and then they have little structures like this. I'm not a very good artist here, but this what we're looking at here is the tail end of the neuron. So this part right here and these little structures coming out, you see that as like this end here. And so neurons look very different than this neuron. What's going on here is it's communicating with other types of cells. These long structures right here are individual skeletal muscle cells. So these are your muscle cells. And this neuron is communicating with it. And the neuron is what stimulates the muscle cells to contract and therefore you to be able to move your skeleton. So skeletal muscles look very different. They're more fibrous and long than a neuron. And the reason why they have these different structures is because they have different functions. And so they're specialized in what they do. So not every single cell looks identical. So when we look at cells and to study them, we need to be able to use tools to see them. They're too small, usually too small to be seen by the naked eye. And that's what this picture is showing you here. That it's showing you, here's the scale of size, largest to smallest. And so it's giving you some things that you can see with your unaided eye. So there are some neurons or nerve cells that you can actually see with your naked eye, not all of them. And then light microscopes. Light microscopes are the microscopes we use here in school. And so they have magnification and it beams light at it. The light shines through the specimen and you're able to see the specimen underneath the light microscope. And so we can see cells, we can see bacteria, some organelles, some larger organelles when we look at it. And then there's an electron microscope. Electron microscopes beams electrons at the specimen. We do not have an electron microscope here, but you're able to see, obviously, you can see here some smaller things. So we can see smaller bacteria. We can start to see some of the macromolecules, proteins, and lipids here, and so on. So we're able to see smaller things with the electron microscope. The electron microscope, there's two kinds. I just want to show you here because your book will show different pictures like this. Transmission beams electrons, it's called transmission because the electrons actually are transmitted or go through the specimen. So that's really good because it lets you see the inside of the specimen that you're looking at. Whereas scanning electron microscopes, the electrons don't go through the specimen, they scan the surface. So you can see the surface of the specimen. This is a cell that has cilia, which are little hair-like structures, and you can actually see the three-dimensional shape of that cilia here. but notice that you can't see the inside. So these two kind of work together. You can do both of them to see different things. Then we use different fluorescents and dyes and things like that to be able to see smaller parts and so on. So that's some examples there. And I put here a link to a website that just gives you an idea of how small things are in relationship to things that you're familiar with. So, so if we look at this, what this does is show some things that we may be familiar with: the coffee bean, Times New Roman font on the 12-point font to give you an idea, a grain of rice, sesame seeds. And then what you do is you take this button down below here, and as you move it to the right, it goes in a little bit, and it starts to show you, okay, now here's a grain of salt, so much smaller than a sesame seed. And then we start to see some cells. So the amoeba and paramecium, those are unicellular organisms. So compared to the grain of salt here, they're smaller. They live in lakes and ponds and fresh water. They're a unicellular, free-living organism. Then look at here, we have the human egg. So this is found in the ovaries of humans. Compare the human egg cell to the other reproductive cell in humans, the sperm cells. Sperm cells are very, very small compared to the egg cells, but they're both part of the human body. Here's your skin cell. This photoreceptor is also called a rod, is found in your eyes to help you see light. And then we have our red blood cells, yeast, and here's your chromosome, X chromosome, which is found in cells. Now we start to see bacteria. So notice here the bacteria, how small it is compared to red blood cells, compared to your skin cell, and so on. Bacteria are very, very small. If we start to look at some of our organelles, notice the bacteria is actually here a little bit smaller than a mitochondria. So it's very small compared to our cells. Then we look at viruses. Viruses very smaller yet. So here are some different types of viruses. And then we start to get into molecules. So here's a ribosome, here's hemoglobin, much smaller, tRNA, antibodies part of your immune system. And then here's some of the ones that we talked about: phospholipids, glucose, adenine, adenine makes up what? What is adenine? It's one of the bases in nucleic acids. That's the A in nucleic acids. Methionine is an amino acid, so we have smaller molecules. And then lastly, we see the water molecule, and lastly, a carbon atom. So it just gives you an idea of just how small some of the things, when we talk about bacteria is really, really small compared to everything else. You can't even see them on this particular picture. So I just thought it was interesting to give perspective about cell size.
So like when you were at like the human egg and like all that stuff, like did you be able to see it with your eye? And then is that
No, usually to see it, you need a light microscope. Yeah. So. Okay, so let's look at the different types of cells. So there are two types of cells: prokaryotic and eukaryotic cells. Eukaryotic cells have compartments. So the compartments are the organelles. So for instance, you've heard of the mitochondria, yes? All right, and chloroplasts. All right, these guys do different functions. So the mitochondria does a process called cellular respiration. Chloroplasts in plant cells does what process? Photosynthesis, right? And so these compartments make the cell efficient, because let's look at the chloroplast, for instance. So all the stuff that's needed for photosynthesis is contained in this compartment, in this chloroplast. So therefore, it's very efficient that chemical reaction of photosynthesis, all the stuff is right there, and therefore it can do photosynthesis at a nice rate. Versus if all the stuff for photosynthesis was just floating around the cell, that wouldn't be a very efficient process, because all the stuff would have to come together. That would be highly unlikely. So having the organelles just is like a special room in the cell, so that you can do a specific function, and each room does a different thing. So, looking at cells of all organisms, there's two types: prokaryotic and eukaryotic cells. It says only organisms of the domain bacteria and archaea consist of prokaryotic cells. All the other cells are eukaryotic cells, so protists, fungi, animals, plants, Let's talk about that. Well, let's talk about these things called domains. Have you guys heard of that? I don't know if they teach that in bio. All right, when we talk about classification of living things, so all living things on Earth is one big group. And then they divide all living things into 3 groups. They call it a domain. So there's the domain bacteria, the domain archaea, and the domain, let's put it over here, the domain eukarya. And so, and they divide all living things into these 3 categories based upon differences. So organisms in this domain have certain things in common to put them in the same group, but they have things that differ from this one and this one, all right? So, and then the same thing with each one of these domains. So then these guys here, what this is saying, the organisms in the domain bacteria and the domain archaea are prokaryotic cells. And the domain eukarya, hence the name, you can assume, is eukaryotic cells. So we go in which category? Eukaryotic cells. So it's telling you here, this last bullet is telling you what kind of organisms would be in that domain. So protists are unicellular organisms, like so that amoeba and the paramecium would be protists. Fungi, mushrooms, yeast. Animals, we would be clumped into that, and plants. And so that's how living things are broken down. So let's compare prokaryotic and eukaryotic cells. And I have a chart here to do that, and I can't figure when we compare and contrast them, we'll look at the differences between them. But there are some similarities. So basic features of all cells. No matter if they're prokaryotic cells or eukaryotic cells. All cells have a plasma membrane. That's another way to say cell membrane, plasma membrane. They have a semi-fluid substance called cytosol. Another way to say that for this class here is cytoplasm. That's what you probably learned. All right. Just think, for all intents and purposes, cytosol and cytoplasm, you can use them interchangeably. There's technically some differences, but for this class you don't need to know those. There's chromosomes. Chromosomes are made up of DNA and carries your genes to code for your proteins. And all cells have ribosomes, which is the site at which proteins are made. Oh, I forget, is this one in here? It's left on my desk. Okay, so I meant to have this chart actually come up one by one, but apparently it's too difficult for me to figure out. So I'm just going to let you copy it down, and then I'll just talk about it briefly. So comparing and contrasting them. So big difference, one has a nucleus, one doesn't. So the eukaryotic cells have the nucleus. We just wrote about things that all cells have in common, and one of those is chromosomes, which contain DNA. So for eukaryotic cells, then that chromosome and DNA is found in the nucleus, but for prokaryotic cells, they have this nucleoid region where it's not doesn't have a membrane around it. It's just a region of the cell where you can find the chromosome and the DNA. And then prokaryotic cells do not have any membrane-bound organelles. This is a key word here. Membrane-bound organelles, which means that they have a phospholipid bilayer around it. So notice that all cells, going back to this, all cells have ribosomes, which make proteins. That is not a membrane-bound organelle. So therefore, prokaryotic cells have ribosomes, but they don't have things like mitochondria, Golgi apparatus, endoplasmic reticulum, things like that. And then their cytosol is bound by a plasma membrane. Since they don't have a nucleus, a lot of bacteria cells are kind of oblong shaped like this. And so the cytosol or cytoplasm is just within the plasma membrane, and it's all within there. And so then they would have a nucleoid region where the DNA is found in there. There on the flip side, the cytoplasm in eukaryotic cells, so this is your eukaryotic cell, since they have a nucleus, here's the nucleus and here's your plasma membrane, your cytosol is in between there, and so therefore fills up all that space. So that's what that means there. And then as we saw in that website and the picture, is that eukaryotic cells are generally way bigger than bacteria. When we look at eukaryotic cells underneath the light microscope here in school, you can see the cell, you can see the nucleus usually. Sometimes it's hard to see the organelles and so on, but you can definitely see the nucleus in the cell. When you look at prokaryotic cells underneath our microscopes, they look like just little dots there. You can't see the inside, you can't see anything, but you can tell that something's there. All right, and so because they're too small, we can't enlarge them enough or magnify them enough with the light microscope to be able to see them clearly. All right, so eukaryotic cells much bigger. And so this is a generic picture of a prokaryotic cell, bacteria. And so here we work from the outside in. There's a capsule and these hair-like structures. We'll talk more about that later in another chapter when we talk about bacteria a little bit more. But right underneath that is a cell wall. So bacteria has cell walls. Our animal cells do not have cell walls, but plant cells do. Fungi do, and so do bacteria. So they have a cell wall. Underneath there is the plasma membrane. There's all the cytosol. This is the big bacteria chromosome. This is the DNA that's in this chromosome. Bacteria have one chromosome, and it's circular. So if I were to actually take this out, if I were to extract this from the bacteria, unravel it, and make it into the big circle, it'd actually be much bigger than the actual bacteria cell. It's very, very big, and so condensed into the bacteria cell. That's different than our cells. Does anybody remember how many chromosomes we have in our cells? Do you remember? No? Way lower than that. I heard 23. That was a popular answer last hour, but that's not correct. Somebody said it over here. No, 46, absolutely. 46 chromosomes. If you said 23, you're probably thinking, we'll talk more about that when we make egg and sperm cells have half the number of chromosomes. They have 23. So normal cells that are not reproductive cells have 46 chromosomes. And they're linear, which means the chromosomes are straight, versus in a bacteria it has the one big circular chromosome. All right, and so that's in that nucleoid region, and then all the little dots are the ribosomes. So basically, they have the DNA contains instructions to make proteins, and they have the spot that proteins are made, which are the ribosomes, and they can do that. And then some bacteria have flagella. Flagella are like little tails that move to help them to move from one place to another. And this is just the real picture, the transmission electron microscope picture that shows those various things. All right, so let's start to look at the parts of the cell. So the plasma membrane. selective, key here, selective barrier, meaning it only allows certain things through it and other things not. Allows sufficient passage of, and these are the things that can go through, oxygen, nutrients, waste, to service the volume of every cell. So these are good, oxygen and nutrients like glucose and things like that. The waste is getting things out, so that we do produce waste, and so they move out. So here's your lipid bilayer. We talked about this last chapter with the phospholipids. So here's your phospholipid bilayer. And let's say this is outside the cell and this is inside the cell. So this is outside and the interior inside. Outside and inside is a what-based solution? Water-based solution. So there's water inside and outside, which remember is why there's a lipid bilayer in the first place. Remember, let's review the phospholipids. This part right here that says this part right here is hydrophilic. So this is, what part of the phospholipid is that? Yeah, it has the phosphate group, right? And so we call it the head. The phosphate group is polar, and so this is the head of the region. It has the phosphate group and is polar. And then the interior is hydrophobic. Why? Because what are these? These are the fatty acids, right? And the fatty acids are, these guys are fatty acids, are made out of basically carbon and hydrogen, which makes it nonpolar, which means it's hydrophobic. All right, so just a quick little review there. So that's why we have that lipid bilayer and why it's constructed that way. So notice that the parts here of the lipid bilayer go with either hydrophilic or polar or nonpolar in the interior. Let's look at the second major molecule in the cell membrane, and these are proteins. So you can see that there are proteins embedded in the membrane, and so there are proteins that go all the way through the membrane, like these guys here. There are proteins that are just on the interior surface here. They don't actually go through the membrane. But what I want to point out is, and I'm going to put kind of a dot here, and dots, and same thing right here, because I want to point out that the proteins follow the same part here. So hydrophobic, so therefore this part of the protein is hydrophobic. This part of the protein is hydrophilic, so it goes in that same region. So hydrophilic, hydrophobic there. And this would be hydrophilic again. So proteins are what? Chains of amino acids, right? So the amino acids that make up the center part here, so if I were to draw, kind of relating it back to last chapter, so here's your chain of amino acids here, here we go, and so on and so forth. So this is to give it this shape, this is your chain of amino acids. The amino acids in this part that's hydrophobic, what would be true about them? What makes amino acids different from one to another? The R groups, right? And so if you're in a hydrophobic region, those R groups are probably hydrophobic, all right? And so therefore that's why this would be hydrophobic here. So an R group that's hydrophobic would be carbons and hydrogens, okay? And so then the amino acids that are over here would be polar amino acids, and so on. So, so that's your proteins. Here they're just showing you, this protein would also go through the lipid bilayer. They're just showing you the top of it there, so it's sticking out. If we looked at this top here, it would also be sticking out. And one other thing, notice that this protein, it has this, what they're trying to show you is it's like a tunnel region through there. And that's because proteins can serve as a way to get things in and out. So certain molecules can go through that tunnel to go into the cell or out of the cell. And the last part is the carbohydrate side chain. So these are carbohydrates. Notice these are rings. So carbohydrates, what's each ring? a monosaccharide, all right, right? Remember, all carbohydrates are either one monosaccharide or a chain of them. So each ring is a monosaccharide, and notice that the side chains can vary depending upon what monosaccharide there is, the chain of it, and so on. And so an analogy I like to use with the carbohydrate side chains is that they are kind of like ID tags. It's a way to tell one cell from another. So within an organism, different cell types can have different carbohydrate tags on them. Or between organisms. So for instance, that's one way that if you have something foreign that comes into your body, let's say a bacteria, there are different carbohydrate side chains on those cells than there are on your cells, and it's a way for your body to recognize foreign particles versus particles that are part of you. And so, so that's what they're kind of used for. So let's look at now the size of cells. We know we just looked at that cells are really small. And let's look at why that is. So it says metabolic requirements. So these are things that you need, the cell needs to survive. So what would it be a metabolic requirement? That's like oxygen, glucose, all right, things that are needed, water. Set upper limits on the size of cells. So upper limits, this means that, so we're made of many smaller cells. So I want to talk a little bit about why we're not made out of just like a few hundred bigger cells. Why do we made up of billions of smaller cells? And one reason is for specialization. So we talked about the neuron versus the muscle cell and so on. And so we can have specialized cells to do different functions. But another reason, a big reason, is these metabolic requirements to get things into the cell at an appropriate rate. So the surface area to volume ratio is critical in one of the big reasons why cells are small and not big. And so what I'd like you to do is take a look at the third sheet of paper that you got here. It says calculating surface area to volume ratios. What I would like you to do is the beginning of it, the top part of that page, talks about surface area, it talks about volume. And then on the bottom, it gives you some generic fake cells that are cube-shaped. And so you have cells A through D. And it tells you how to calculate surface area and volume of a cube. And so what I'm going to want you to do is read through that and then go through and actually figure out the surface area to volume ratio for those 4 cells, A through D, and fill in that chart at the bottom. All right? For that, you will need a ruler. I put a bin of rulers up here that you can constantly get, all right, when you get to that point. But read through the background information first. This is one of the things on the math section that you could be asked to do on the test. Is to calculate surface area. probably have like a 180 degree angle this way. I'm gonna wait. Ask him. Yeah, it's
Is that thing homework?
the focus questions.
Yeah.
There's just 5 of them.
Okay.